Excellent. I promised you an example of a titration experiment, and this is a very interesting one. It is actually to determine the concentration of ethanoic acid in vinegar. It was found that 40 milliliters of 0.5, and I forgot to put this in, of sodium hydroxide, 0.5 moles per cubic decimeter, that should be in there, of sodium hydroxide neutralized 20 milliliters of vinegar. Okay, so how was this experiment done? It was done using a pipette to drip in here into the burette. We had our known concentration. This was our known concentration of sodium hydroxide. So sodium hydroxide was known, and this concentration was 0.5 moles per cubic decimeter. And what we also find is that in here we have 40 milliliters used. So at the end of the experiment we had used 40 milliliters was our volume. So our volume was 40 milliliters. And here's now important point to remember with volume. We have to work in liters because 1 liter is 1 cubic decimeter. Important. So in order to get to liters, please remember milli means I have to divide with 1,000. So I divide with 1,000, and that now gives me 0.04 liters or cubic decimeters. Okay, that's how much was used. This was dripped into our vinegar. Now vinegar, as you know, is a clear solution. And first draw my conical flask. So there's my conical flask. In my conical flask I've got a clear solution of vinegar. Oh, that's not clear. Okay, a clear solution of vinegar, but it should not be clear because I want to neutralize it. I already suspect that it has acid in it, so what we add is some methyl orange, and if we add methyl orange in here, it has a reddish color. It has a reddish color. And now we drip sodium hydroxide in here so that the reddish color becomes orange. Okay, in other words, we're going from less than 4.5 up to about 6, and at 6 it should be yellow. So we're aiming for yellow. In between we're going to have an orange color, but once it gets to yellow, then we know that we're at more than 6, and we won't go much higher than that. Okay, and what we found here, we knew that this was 20 milliliters. So the volume of the unknown volume was 20 milliliters, because that we can easily measure. And we want to know how many moles were in here so that we can use the unknown moles to find the concentration of acetic acid. Now before we can go any further, we need to actually find the balanced equation. So ethanoic acid reacts with sodium hydroxide. Ethanoic, okay, tells me there's 2 carbons. Ethanoic, so here's my 2 carbons. Okay, I know 1 carbon is double bonded with oxygen because ethanoic, and single bonded with hydroxide. This is the carboxylic acid. Okay, that's a carboxylic acid. We added an extra carbon because we need 2 carbons, and this carbon must then just be filled up with hydrogens. Okay, so here's my ethanoic acid. That is carbon with 3 hydrogens, carbon double OH. That's the signature of carboxylic acids, that COOH, COO, plus sodium hydroxide, and this is supposed to produce for me a salt. okay, a salt. Now you might not know what the salt is. Well, let's do this. Let's just, we know it will produce salt and water, isn't it? Okay, so if we used this to get the water, we must have just used another hydrogen here. Okay, so if we take away this hydrogen, it will leave us with this, whatever that is called, it doesn't matter, but we know that this was our H plus, and this was our cation, is it? No, anion. This was, no, it was my cation. This was positive, which makes this one negative. This one is one positive again. This anion was one negative, so we will have our sodium, and then our cation, CH3COO. Now, technically correct way to write this is to put this one at the end. Okay, you might just recall that we always write our carbons and hydrogens first, but it's not the worst case in the world. This is not what we're trying to find. As a matter of fact, this question comes from a past exam paper, and they actually gave you the equation. Okay, so that's nice of them. Let's just see if we need to do any sort of balancing act in here. We already have, just look at our ions. Here we have this ion completely represented there, so don't even go and count them separately. It's completely represented there, completely represented there. One sodium. My hydroxide and another hydrogen produces water, so this is great. Every 1 moles of ethanoic acid will react with 1 mole of sodium hydroxide, which is awesome because it makes this thing so much easier. So all I need to know is, if I want to know how many moles of unknown there was in here, all I need to do is find out, well, how many moles was used. I have my volume and I have my okay, so let's work out moles used. Okay, so moles used would be, well, my N is equal to my concentration times my volume, which my concentration was 0,5 and my volume was times 0,04. Okay, so this is basically a half of that gives me 0,02 mole. That is how many moles of sodium hydroxide was used to neutralize 20 milliliters of ethanoic acid. So if this is how many moles I used, the moles of acid reacted is also equal to 0,02. Why do I say it's also equal? Because it's a 1-to-1 relationship. For every 1 moles of this one, there will be 1 moles of that one. So since they are 1-to-1, it will be the same. If this was, let's just use an example. If this was, for example, 2, then I know that the moles used have to be double than the moles that was the unknown moles, which, so if this one was 0,02, this one was going to be 0,01. The relationship between these two must be the same as the relationship between those two. Okay, so I know the moles reacted was that, so all I now go and do is say, okay, well, what was the unknown concentration? The unknown concentration is was my unknown moles divided by the volume of the unknown substance, which in this case is 0,02 divided by the volume here. Okay, again is in milliliters, so we need to divide with 1,000 to give me 0,02 cubic decimeters. Excellent. Oh, isn't that a lovely answer? Okay, there's 1 mole per cubic decimeter of ethanoic acid, according to this experiment, ethanoic acid in vinegar. I think that is a great result. I hope it made sense to you, but actually in the previous video I gave you a little formula. Let's see if I can remember that formula. The concentration of the unknown is equal to the moles of the unknown divided by the volume of the unknown. Okay, but the moles of the unknown we found by taking the concentration, the moles of the known. Okay, the moles of the known, which is the concentration of the known, that's not with an N, known, multiplied by the volume of the known, divided by the ratio, that's this R, the coefficient here, the ratio of the known, and it was multiplied by the ratio of the unknown, and this whole thing was divided by the, that was just this value, so the whole thing must just still be divided by the volume of the unknown, so volume of the unknown. So there's basically my formula. And now if I wanted to just use this formula to get the concentration of the unknown, I will simply substitute all of my values. What is the ratio of the unknown? The RU and RK is, RU is the coefficient here of the unknown value, which is 1. So it's 1 times the concentration of the known value is 0,5. Okay, that was the concentration of sodium hydroxide known. The volume of sodium hydroxide was 0,04 when I simplified it. When I converted it, divided by 1, which was the ratio or the coefficient of the known, that's that one, times the volume of the unknown, which was 0,02. Let's see if it gives me the same answer. Yes, it does. 0,5 times 0,04 is basically half of 0,04, which gives me 0,02. 1 times that keeps 0,02. Divided by 1 times 0,02 is exactly the same as the numerator, which gives me an answer of 1 mole per cubic decimeter. Excellent. Thank you very much for watching. I hope this was helpful and that you can see it's really not as intimidating as it seems. Thank you, and we'll see you in the next video.